when we put all the enzymes that we've learned in action together, DNA replication looks something like this. Helicase will begin separation of the parental DNA, single-strand binding proteins stabilize the strands, and primase and polymerase will work their way down the strand to synthesize new DNA. To put all this detail into a bigger context, it's important to note that DNA strands aren't replicated from one end of the DNA to another in just one fell swoop. There are actually multiple sections of DNA being replicated at the same time within a strand. And as the DNA strands are separated by helicase, these bubble-like structures sort of form in the DNA, and they sort of, in a very simplified depiction, look like this. And so what's happening, in one strand of replicating DNA, there will be several bubbles all along the strand, slowly growing as the enzymes work their way out in opposing directions. And the enzymes will start at the center, which is called the origin of replication, and start moving out in opposite directions along both strands. Eventually, all the bubbles will all join, completely separating the entire DNA double helix as synthesis of the new complementary strands are being completed. So this depiction here, this large depiction, is actually only a close-up view of one section of this bubble. And this section and this section is known as the replication fork. So DNA replication involves some rules. It's not just as simple as these enzymes working together to make DNA strands. One of the biggest rules is that DNA will always and only be synthesized from the 5' to the 3' end. And this is not talking about the template strand. It's talking about the strand that's being synthesized at the moment. So, for example, if we take a look at this strand, we can trace it back to here, and we notice that this end is the 5' end, so that means this end must be the 3' end, and the 5' and 3' end are just referring to certain molecular complexities within the ends of the DNA molecules. And you can see this arrow pointing here shows that the DNA strand is being synthesized from 5' to 3', as we said. But if you were to look at this strand, it's similarly moving in this direction, but if we were to trace back the ends, this is the 3' end, and this is the 5' end. So how exactly is it being synthesized in this direction in a 3' to 5' end when we said that it can only be synthesized from 5' to 3'? Well, despite the DNA traveling in the opposite direction, 3' to 5', there is actually a particular synthesis method that allows the DNA to achieve 5' to 3' synthesis. So the enzymes will synthesize DNA in sections from 5' to 3'. So for example, DNA polymerase will synthesize this section here, and then it'll skip over a section of DNA and start here, and then work backwards. And so now notice when you're working backwards, you're going from the 5' to 3' end. And these little DNA fragments are called Okazaki fragments because they were discovered by a Japanese scientist of the same name. And so that means if you were to look at this bubble here for the overall picture, let's label, this is the 5' end, this is the 3' end, and this is the 3' end, the 5' end. Down here, it's being synthesized, the DNA is being synthesized smoothly because it's 5' to 3'. And up here, again, it's 5' to 3', so it's synthesizing smoothly. But up here, it's sort of synthesized like this, in little chunks. And over here, the same, it's being synthesized like this in little chunks. The strand that's synthesized through Okazaki fragments is known as the lagging strand because it's always slightly slower than the other strand, lagging spelled like this. And the other strand is called the leading strand, which smoothly synthesizes from the 5' to the 3'.